Good morning.
Good morning, sir.
Good morning,
Good
sir.
morning,
Morning,
sir.
sir.
Good morning,
Good
sir.
morning,
Good
Morning, sir.
morning, sir.
Very
Good
good
morning,
morning.
sir.
Morning, sir.
Okay.
Good morning, sir.
Okay. See, today already you have discussed about, you have studied in your 10+2, what is central dogma of molecular biology. So what happens, our genetic characters transfer from DNA to daughters. Hmm. This is already you have studied to understand, before understand the central dogma of molecular biology. The flow of genetic information from DNA to RNA, RNA to protein is collectively called as central dogma of molecular biology. It is formulated by Francis Crick, the central dogma of molecular biology, which compromises three major processes. That is, you can see that already you have seen replications. Replications. You know, earlier, don't make any wording on the ಅವ್ರಿಗೆ ಕ್ಲಿಯರ್ ಇರ್ತದಲ್ಲ.
Clear.
ಸೆಕೆಂಡ್ ಮೇಲ್ಗಡೆ ಇರ್ಬಿಟ್ಟು
Clear.
see that these are the three steps of process of replication, transcription, translations. These three steps involved in the synthesis of protein. So protein is the which has cellular functions, many functions, whatever enzymatic function, immune functions, you can take contractile functions, structural functions. All these have specific characteristic transformations from DNA to protein, which perform specific functions in our body. In the cell, you know, in the cell there will be nucleus. You can take eukaryotic cells. The nucleus contain chromosomes. Chromosomes is having one genetic information component is DNA, the double helical structures. What is DNA? You must understand. Then you will understand the what is central dogma of molecular biology. See, copying of the replica. You know, replica means duplications. Same copies synthesized from DNA polymerase to another daughter DNA. Then this DNA dependent DNA polymerase, so it synthesizes the mRNA. Understood? This mRNA synthesis from DNA to mRNA, that process is called transcription. This is process by which genetic message encodes in the DNA are copied preciously into RNA. Then it is involved in the synthesis genetic encoded in the messenger RNA to translate into the protein and which synthesize the specific protein. This is called central dogma of protein. Before going to understand this replication, transcription, translation, you must know basic aspect of nucleotides. What is nucleic acids? See, this is DNA itself replicates and forms the daughter DNA, encodes, then daughter DNA to mRNA by DNA dependent mRNA transfer RNA polymerase, which synthesizes the mRNA, then mRNA to protein. This is the simple concept of what is central dogma of molecular biology. Synthesis of protein from DNA through three major process of replication, transcription, translations. Details you will study in once you come over here in the molecular biology classes. It is requires many more hours to understand what is replications, how it replicates, what are the codons, what is genetic code, what is gene. Everything you have to go through details in the what is replications, then transcription, post-transcription modifications, then translation, post-translation modifications, and how it's synthesized. The chromosomes contain specific codes which synthesis. Suppose insulin you want to synthesize, there is present in the insulin uh, codes in the chromosomes, then it synthesizes the insulin. Let us see, you have to understand nucleic acid, then you will understand how protein is synthesizing. Already we have studied in your undergraduate in 10 plus 2. See, you have to see the first, you have to describe the structures of nucleotides, then illustration of biological importance of nucleotides, their importance, major three types of synthetic analog of nucleotides and antimetabolites, their clinical significance. Then you have to study, describe the structure and function of DNA, then explain how DNA is organized in the cell, that you must understand in the chromosomes. Then it codes for the synthesis of protein. See, nucleic acids are macromolecules present in all living cells in combinations with protein to form nucleoproteins. The protein usually proteins and histones. Genetic information is encoded in the nucleic acids molecules. See, nucleic acids are the polymers of, can you see, nucleotides linked by phosphodiester bond. They are therefore called as polynucleotides. DNA is a molecule of sequence of polynucleotides. The nucleic acids are two main types, deoxyribonucleic acids, then ribonucleic acids, or it is called RNA. DNA is present in nuclei. A small amount are also present in mitochondria, whereas 90% of RNA is present in cell cytoplasm and 10% in nucleolus. See, structure and function of nucleotides. Each nucleotide consists three components. A nitrogen base, already you know that, there is six. A nitrogen base, a pentose sugar, phosphate groups. The nitrogen bases of DNA and RNA, RNA and DNA, the two class of nitrogen bases, purine and pyrimidines are bases are present in RNA and DNA. Purine bases, two principles purine bases found in DNA as well as RNAs. Adenine and guanine is common in both the cases. The purine base. Then pyrimidine bases. So you have a purine ring, then there will be adenine and guanine structure. Then pyrimidine bases, cytosine, uracil, and thymine. The cytosine and uracils are found in RNA, and cytosine and thymine in DNA. Here is thymine is replaced by uracil in RNA. So both DNA and RNA contain pyrimidine, cytosine, but they differ in second pyrimidine base. DNA contain thymine, whereas RNA contain uracil. Okay, is it clear? Can you hear?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Yes, sir.
Yes. Okay, you understand these things. Pyrimidine, leave it a ring structure. Then we'll go for there will be sugar is present. The pentose sugar is either D-ribose or deoxyribose. DNA contain 2-deoxy, that is second position, deoxyribose. RNA contain D-ribose. A pentose sugar, D-ribose, deoxyribose are linked a base, purine and pyrimidine, by a covalent N-glycosidic bond. The term is called nucleoside. Is used structure containing only sugar and nitrogen. If nitrogen bases are there in nucleic acids, then it is attached with sugar is called nucleosides. Then this is called an example in sugar. It's having the base, sugar and having the base. See, the nucleotides, which is having another component is phosphate. The nucleosides are adenosine, guanine, cytosine, thymine, and uracil are named. These are respectively. If sugar is ribose, is produced ribonucleosides. If sugar is 2-deoxyribose, that a deoxyribonucleic acids. Structure of you see nucleotides. Nucleotides are phosphorylated to nucleosides. So nucleosides are nitrogen bases containing pentose sugar. The phosphate group is attached to nucleoside by an ester linkage to hydroxyl group of pentose sugar. Already you have studied, just you go through, just you know, deoxyribonucleotides and ribonucleotides. This is the structure of nucleotides. Then you have mononucleotides or nucleosides in which single phosphate group is attached, that is hydroxyl group of the pentose sugar. For example, AMP, that is called single phosphate is there, that is adenosine monophosphate, that is called AMP. If additional phosphate group is attached to pre-existing phosphate of mononucleotides, then it will become ADP, diphosphate, then triphosphate. You know energy component, highly rich energy component is phosphate, is ADP. Say adenosine, all diphosphate, monophosphate, adenosine, triphosphate, adenine groups are present. That's the high energy phosphate bond. Phosphate, high energy phosphate, alpha, beta, gamma positions.
Hello, sir.
Yes, please.
Your voice is not coming clearly.
Okay, okay. Okay. Besides being the structure component of nucleic acids, several nucleotides such as ATP, ADP, these are the triphosphates are formed. According to basis, you know, GTP, GDP, GMP, all mononucleotide to triphosphate components, which have biological functions. Such nucleotides are called biologically important nucleotides. See, ATP serve as a biological source of energy in the cells. ATP is required as a source of energy in several metabolic functions. Metabolic pathway, for example, fatty acid synthesis, glycolysis, several metabolic pathway operates in the cellular level. Cholesterol synthesis, protein synthesis, even gluconeogenesis, HMP pathway, etc. The physiological functions such as muscle contractions, nerve impulses, transmissions, etc. All the functions energy is required. AMP, AMP is the component of many coenzymes, NAD, NADP, FAD, coenzyme A. These coenzymes are essential for metabolism, carbohydrate and lipid proteins. Cyclic AMP is found from the ATP by the action of adenylate cyclase, which is the intermeter of second messenger. So for many hormones, epinephrine and glucagon. Cyclic AMP affects a wide range of cellular process by the active second messengers. These things all cellular reactions, what are the energy, how energy requires, how it is adenylate cyclase, protein kinase involved in this process. It enhances the degradation of storage of fuel like fat, glycogen by stimulating glycolysis and glycogenolysis. So it inhibits the aggregations of blood pressure, etc. All these are the functions. Cyclic AMP increase the secretions of acid by gastric mucosa. See, these are hormone using where cyclic AMP as a second messenger of synthesis of all these components. Then GDP, and then you go for GTP. All these things, you know, how it involved in the cyclic AMP, GMP in the various metabolic functions, anticoagulative functions to cyclic AMP. So UDP participation in glycogenosis, details you will study in the once you join here, the particular topic, they will go stepwise. See, these are the many functional role of DNA involved in the metabolic functions. And also there will be synthesis analogs of purine pyrimidines are using the medical treatments. They use by chemotherapy to control cancers and infections. So these are the analogs of purine pyrimidines. used for treatments. 5-chloro, 5-iodo, uracil, what you know, mercaptopurine, and 5,6 azouridine, etc. So next we can go for several analogs used for medical treatments. Then we'll come to the what are the DNA, how it structures, what are its functions. See that DNA already you know DNA serve as a it is present in the it is involved in the synthesis of protein, which is present in used in the prokaryotic and eukaryotic cells. In eukaryotic DNA located in nucleus from cytoplasm and nuclear membrane. Because of prokaryotic lack in the internal membrane system, their DNA is not separated from the rest of cellular content. DNA is present in mitochondria and chloroplast, etc. Many viruses also contain DNA as their genetic materials. Structure of nucleic acids is described in terms of primary, secondary, and tertiary and quaternary levels. So, primary structure of DNA, deoxyribonucleic acids, is a polymer repeating subunit called deoxyribonucleotides. In the polymer, nucleotides are joined one another by 3' 5' phosphodiester bonds. The primary structure of nucleic acid refers to the sequence of its nucleotides residues. See, each deoxyribonucleic acid subunit is composed with three parts: 5-carbon, pentose sugar, deoxyribose sugars, nitrogen base, purine, and pyrimidine bases, then phosphate groups. So then you have hydroxyl group, all these structures, you can see that. A backbone of DNA consisting many deoxyribonucleic covalent 3' 5' phosphodiester. The sequence basis along with polynucleotide chain is not restricted in any way. Precise sequence based on carries or the genetic information. These are the structures of DNA, how base, sugar, and how it is polypeptide nucleotide is linked, backbone of the structure of DNA. A second structure of DNA, in the DNA, second structure relates to the helix form by interactions of two DNA strands. It consists two helical DNA chain would around the same axis to form right-handed double helix. The chains are paired in an anti-parallel manner, that is 5' end and one strand paired with the 3' end as the other strand. There are two strands. So one is having a 5' 3', other one is parallelly 3' 5' strands. So there will be hydrophilic deoxyribose and phosphate groups, backbone of each chain, or all the whatever characteristic properties of DNAs. So see here, there will be base pairing of nitrogen base pairing, adenine always based with thymine, and guanine based with cytosine. This is the the Chargaff rule, how it is held by double bond and triple bonds. This is a structure of double helical structures of DNA. One strand DNA are paired with the basis of the second strand to the adenine. All this is what Chargaff says, that adenine is paired with thymine, guanine is paired with cytosine. So either one bond or double bond or triple bond, one polypeptide chain of DNA double helix, always component to the others. Hydrogen bonding. This is hydrogen bonding, base pairing between adenosine, adenine and thymine. These are DNA's very flexible molecules, ability to exist various form of based on the environmental conditions, future known as structural polymorphism. Then three different types of structural component of DNA, then aDNA, bDNA, zDNA. There will be, see that right-handed, even this all bDNA, zDNA. There are the sequence of nucleobase group C's varies from one to other. See all the informations in this slide. What is DNA? DNA right-handed, one is left-handed, and angstrom diameters, number of base pairs per helix, 11.6, 10, 9, 12, so on. And this is a Chargaff rule, very essential to remember. The basic composition of DNA generally varies from one species to another species. DNA specimen isolated from different tissue of the same species have the same base composition. The base composition of DNA is given. A species does not change with the an organism, age or nutrient status or changing any environmental status. You can see adenine residue is equal to the number of thymine. A is equal to T. Number of guanosine residue equal to the cytosine residue. From these relationships follow that sum of the purine residue equal to the sum of the pyrimidine residue. A plus G is equal to T plus C. The ratio of purine and pyrimidine bases of DNA is always 1. These quantitative relationship is sometimes called as Chargaff's rule. Watson and Crick deduced the adenine must be pair with thymine and guanine with the cytosine because steric and hydrogen bonding factor. Adenine cannot pair with the cytosine. Guanine cannot pair with the thymine. Thus, one member of base pair in a DNA must always be purine and other is pyrimidine. This base pairing restriction explain the double standard DNA molecules, a content of A equal to the T and G is equal to the C. And there will be tertiary structure of DNA or nucleic acid tertiary structure refers to the overall three-dimensional shape. The DNA tertiary structure arises from the supercoiling, which involve double helical helix being twisted into or tighter and more compact shape. Then similarly, quaternary structures, this DNA is connected in histones, non-histones proteins from the chromatin. The negative charges due to phosphate group in a DNA makes its relative only acidic. The negative charges binds to the basic histone group. Then a eukaryotic cell is not actively dividing. Its nucleus is occupied by the chromatin, which it is compact from the DNA twisted around the protein known as histones. And all characteristic properties of chromatin structures. So this is chromatin in non-dividing cells. So chromatin in cell prepared for divisions. Function of DNA, stored genetic information. The information is stored among 46, 23 pairs of chromosomes. These chromosomes are made up of thousands of shorter segments of DNA called gene. Gene is the segment of DNA polynucleotides. Each gene stored directions for synthesis of all protein molecules of the cell. See, here is the how the protein functions, how protein is synthesized from the gene. Gene is which is a segment of DNA. From DNA to replicates, then RNA, RNA to this is DNA to mRNA, then mRNA to translate to the protein as specific in the cells. Protein act as an enzyme. All what are the proteins are synthesized, which are become a functional protein. Which are functional proteins are act as enzyme, structural components, hormones, and various other functional molecules. Eukaryotic DNA organized into chromatin. Eukaryotic DNA tightly bound all the characteristic properties of this DNA in the chromatin. Our structure, chemistry of nucleic acid, you will study in one's classes. They will take in the basic aspects in routine classes. See all the number of characteristic properties, 4 major bases of RNA, purine base adenine and guanine, cytosine and uracil is there in RNA. The difference of RNA and DNA, RNA is sugar, is ribose in DNA deoxyribose. RNA does not possess thymine except in rare cases. Instead of thymine, RNA contain uracil. This is a major difference in the RNA and DNA. the bases differ. In the RNA, adenine base with uracil rather than thymine. See that next is cell contain three major type of RNA. What it is involved in the synthesis of protein, messenger RNA, transfer RNA, and rRNA, all individual in the process of protein biosynthesis. Each differ from other by size or functions. Structure of mRNA, mRNA cover 5 to 10 all the characteristic properties of mRNA synthesis from nucleus. Then mRNA carries the genetic information from its codon, and then eukaryotic cell characteristic by example pi end of mRNA copied by the 7-methyl. All these details into it. Then you can go functions of mRNA. mRNA serve as a template for protein biosynthesis. What we'll see in the central dogma of protein synthesis from DNA to mRNA. So this is the role of mRNA. mRNA serve as a template for protein biosynthesis. Transfer the genetic information from the DNA to protein synthesis machinery. If the mRNA codes for the only one peptide, the mRNA is monocystomic. If it codes for two or more different polypeptide, the mRNA is polycystomic. So in eukaryotic cell, most of the mRNA is monocystomic. See, these are the structure of mRNA. See, tRNA, and that one, molecules vary in the length from 74 to 95 nucleotides. In eukaryotic cells, all these characteristic properties of tRNAs. So there will be component tRNA, dihydrouracil, in which one double bond of the base is reduced, so on. Thymine, pseudouridine, which uracil is attached to ribose by carbon-carbon bond rather than nitrogens. So structure of tRNA, see all single standard transfer RNA molecule gets folded into the structure that appears like a clever leaf model. Usually it is important question usually asked in right note on tRNA. All tRNA contain 4 main arms. These arms you have to write acceptor arms, D arms, anticodon arms, TCI. C arm is the clever leaf structure of tRNA. This three-dimensional structure of tRNA. Then explain the whatever functions of acceptor arm, D arm, anticodons. So TCI arm. Then function of tRNA. tRNA carries the amino acids in activated from to the ribosomes for protein synthesis. See, RNA, ribosal RNA is called ribosomes, where protein is synthesized, is called rRNA, ribosome, the cytoplasmic nucleoprotein that act as a machinery for the protein synthesis. See again, prokaryotic, eukaryotic cells, ribosomal, subunit 40 and 30 subunits. See, eukaryotic ribosomes composed of 60S and 40S subunits. Then function of ribosal RNA. The function of ribosal RNA molecule is the ribosomes. Particles are not fully understood, but they are necessary to maintain a ribosome structures and also participate in protein synthesis binding mRNAs. Okay, now see that. Next, see what is next is what is the central dogma involved in the process involved in replications, proofreading, describe the end replication problems, eukaryotics, describe the process involved in repair of DNA. These are the objective. Name inhibitor of DNA replications, give there its clinical importance, outlines of cell cycles, know what is meant by chromosomal recombinations. So all these things we'll study in the regular classes. See again a central dogma, what you have the flow of genetic information DNA to RNA to protein in living cell is called central dogma of molecular biology. There are replications, transcription and translations. Then you should know that details we will study in the next classes, replications. What replication, duplication of synthesis of DNA is called replications. DNA
Sir, you are
replication is,
repeating the slides.
please.
You are repeating the slides.
ಏನಂತ maybe internet problem, huh? Just you see, duplication of synthesis of DNA is called replication. So we'll study details in once you join to the regular classes. Just it is the information, basic informations. What is central dogma of molecular biology? How protein is synthesized? There is a three process of replications, transcription, translation. Each has it requires 2 hours to understand what is the replications, how genetic information is transferred from coding from the nucleotides to amino acids by DNA polymerase, RNA polymerase, so, and how a specific protein is synthesized. So we'll study in the given time. This is just to go through just for your informations. What are the things you have to study in the uh, central dogma of molecular biology. We will not understand whole protein synthesis in this given time of 40 minutes. Just basic, you can go through with the nucleotide, nucleic acids, nucleotide, nucleosides, and what are the base pairs and what is mRNA, tRNA, all details you have to study in one by one. Then you will understand what are the protein and how protein, specific protein is synthesized by coding by codons. Then you have to understand gene, genetic codes, and all codons, so, and they're used in the protein synthesis. Say this is C replication is semi-conservative replication. C stands serve as a template. One act as a template for synthesis of mRNA. They produce the 2 new DNA molecules. With one is new strand, one is old strand. The new strand is from and uh, complementary to each other, like the parent DNA. Then it is semi-conjugated to daughter DNA, 2 DNA. One is comes from parent, other one is new strand is formed. Again, basic requirements for the replication substrate, template, primer, enzymes, proteins, which involved many processes in the formations of protein synthesis from the DNA. First is replication also itself involved these many basic requirements. Then you should have the substrate, all deoxy, adenosine triphosphate, deoxy, all guanosine triphosphate, cytosine triphosphate, thiamine triphosphates, all are basic substrate required for this process. Then template is required for direct additions of appropriate complementary nucleotide. Complementary nucleotides you have to involved in the synthesis of DNA strand. Each strand is parent double helix, act as a template for synthesis of new strand. So go on, one cell is converted into new cells, another cell synthesized. Similar way, it is the DNA synthesis the daughter DNA, then it synthesizes the mRNA. So there will be requires for primer for even mRNA synthesis. DNA synthesis cannot initiated without primer. A primer is short piece of RNA. RNA synthesis is not possible without primer of RNA in the DNA strand. RNA polymerase, which is the synthesize the short stretch of RNA that is complementary to one of the template of DNA strands. These are primers, role of primers. So how it is the 3 prime, 5 prime end primase, the RNA primase is requires the 5 prime, 3 prime. Then it forms the complementary of the double helical structure of newly synthesized DNA, which is a new strand and old strand replicates. So we'll study details next whatever classes we take in the molecular biology. So we'll go according to in systematic way. So I'll synthesis of an enzyme involved DNA polymerase, RNA polymerase. So all these enzymes are involved in the synthesis. Stages for initiation or elongation, so termination are only in the replications. Each what is initiation, what is elongation, terminations, we'll study in the protein synthesis. See, we will not go into details of initiations. We'll study in the routine classes. What is initiations? What is the origin? How it replicates, getting the fork. This is how it's synthesizing the duplicating the daughter cells, genetic information from parent to daughter cells. So there will be fragments are there, that fragments are cause of the fragments. So all these things we'll study in the once you join here. Okay. So only today class, what is central dogma of molecular biology is synthesis of protein from the DNA, genetic information transfer from the DNA to RNA to protein through three steps of process, replication, transcription, and translations. Okay. The little bit. ಏನಾದ್ರಿ ಇದು ಮೇಜರ್ ಟಾಪಿಕ್ ಇದು ಇಡಬಾರ್ದು. HOD ಕ್ಲಾಸ್ ರೀ ನಮ್ಮ ಒಳಗೆ ಬರ್ತಾವ. ನೆಕ್ಸ್ಟ್ ಒಳಬೇಕಾದ್ರೂ ನಾನೇ ತಗೋಬೇಕು.
ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇದ್ರೊಳಗ